Hello, I am Nima Sharifi from Cleveland Clinic. I'm a medical oncologist and basic scientist, and my laboratory group is focused on steroid metabolic processes that drive prostate cancer resistance to hormonal treatments. The growth of advanced prostate cancer is initially driven by two sources of androgens, gonadal androgens, or testosterone, and extragonadal-derived androgens. Most of the extragonadal androgens probably come from the adrenal gland in the form of DHEA and DHEA sulfate. The long-standing initial systemic treatment for prostate cancer was testosterone deprivation, in other words, medical or surgical castration. However, positive clinical responses are usually followed by the eventual development of resistance as castration-resistant prostate cancer. With gonadal androgens blocked, tumors utilize extragonadal precursor steroids, or DHEA, to make their own potent androgens, such as testosterone and dihydrotestosterone, otherwise known as DHT. The enzyme 3-beta-HSD1, which is encoded by the gene HSD3B1, is essential in the conversion from DHEA to DHT. This process is one of the major drivers of castration-resistant prostate cancer. Several years ago, we noticed that different prostate cancer models have strikingly dimorphic metabolic phenotypes. It turns out a very common germline variant in HSD3B1 encodes a single amino acid change in 3-beta-HSD1. That amino acid change affects the conversion of DHEA to DHT. The wild-type gene on the left encodes an enzyme that is rapidly degraded and therefore has low steady-state levels that lead to low levels of DHT synthesis from extragonadal precursor steroids. The enzyme encoded by the germline variant on the right encodes a stable enzyme that produces much higher levels of DHT from extragonadal precursor steroids. These higher DHT levels stimulate the androgen receptor and therefore disease progression. We now know that inheritance of the HSD3B1 variant is associated with worse clinical outcomes for patients with advanced prostate cancer treated with castration. These clinical findings have been independently validated and now are clear across 6 patient cohorts. Together, this means that the rapid metabolic phenotype encoded by the HSD3B1 variant translates to a clinical phenotype of patients with more extragonadal-dependent tumors. In these patients, castration alone is probably not enough. These patients in particular may benefit from extragonadal androgen ablation. Recently, 2 large clinical trials have shown a clinical benefit from adding abiraterone, which is a drug that blocks extragonadal androgens, to castration up front. These are the Latitude and Stampede clinical trials. From our prior work, we know that 3-beta-HSD also metabolizes abiraterone because of its similarity to the steroidal structure of DHEA. Metabolism of abiraterone by 3-beta-HSD leads to multiple downstream metabolites, one of which is called 5-alpha abiraterone, which is capable of stimulating the androgen receptor and promoting tumor progression in xenograft models. The overarching question for this study in the Journal of Clinical Investigation is whether HSD3B1 genotype regulates 5-alpha abiraterone synthesis in patients. First, we had to make sure we took into account the pharmacokinetics of abiraterone steroidal metabolites. Here you can see the pharmacokinetics of 7 steroidal metabolites after a single dose of abiraterone acetate in 15 healthy volunteers. Next, we assessed circulating concentrations of abiraterone and its steroidal metabolites in 30 patients with castration-resistant prostate cancer. We used the time from last dose of abiraterone to blood draw and the pharmacokinetic data in the first part of the study to normalize for pharmacokinetic characteristics. Of the 7 abiraterone steroidal metabolites, only the 5-alpha reduced metabolites, one of which is known to stimulate the androgen receptor, correlated with HSD3B1 genotype. Here, you see a genotype-dependent stepwise increase in the normalized concentration of 5-alpha abiraterone, which we now know is a modest AR agonist. In conclusion, the HSD3B1 variant drives a clinical phenotype of extragonadal androgen-driven prostate cancer. Thus, men who inherit the variant are probably more likely to benefit from blocking extragonadal androgen synthesis. However, our research shows that when this is done with abiraterone, patients make more of an abiraterone metabolite that effectively acts as a modest androgen, potentially partially reversing the intended purpose in this subset of men. The ultimate clinical consequences of this pharmacogenetic effect of abiraterone metabolism has yet to be determined, and more studies are needed to investigate the consequences on clinical response. Thank you.